Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 3rd February 2021. As all of us know, we are into the revision mode. We are revising entire syllabus in the form of test papers now. Last time around we have done revision through test paper 1. This time we are doing through test paper 2. And the chapter that we are concerned in this session is excretory products and their elimination. So as usual, the purpose of giving this test paper training is to make you familiar with 1 mark questions, 2 mark questions, 3 marks questions, and of course long answer type 5 mark question. This will give you a fair idea about what are the topics that you need to focus in this chapter and what are the topology of the questions, and then most important, how to present your answer. So always substantiate your answers, particularly questions of 3 marks and 5 marks nature. You need to substantiate your answer with a beautiful diagram labeled sketch. So keeping that in our background, here we go. First we go with the questions followed by the answers. Question 1. Name the 3 kinds of nitrogen excretion. So nitrogen form excretion. What are podocytes? Neat exam question also. Question 3. Besides water, name any 2 constituents of human sweat. 4th, kidneys do not play a major role in excretion in amniotic animals. Justify. So that is a statement and you need to justify the statement. Define glomerular filtration rate, GFR. So expected question, what is its value in a healthy human being? Question 6th, what is the significance of frog's tadpole being amniotic, ammonia, and the adult frog being ureotelic? Next, describe urea cycle. This is something new in this chapter. And then again, very much expected question from this chapter. What is a dialysis machine? When is it needed? And maybe how does it function? That's also important. And long answer type 5 mark question, very important question. Define the mechanism of urine formation. This is very much expected from this chapter as long answer type question. Now having read all the questions, now take an answer sheet, start writing the answers. You may take 30 to 40 minutes, and whatever instructions I have given to you, keep that in mind and answer it. After having answered, come back to this session. Now in this I am giving you the answers. Compare your answers with the answers that I am providing with. Question number 1. 3 forms of nitrogen waste: ammoniotelism, ureotelism, uricotelism. So wastes are thrown out in the form of ammonia, NH3, in the form of urea, and in the form of uric acid. Question 2. Answer. Epithelial cells of Bowman's capsule are called podocytes. Very important one. Epithelial cells of Bowman's capsule are called podocytes. Question 3. Sodium chloride and urea. Of course, we know in the sweat, these are the 2 things that come out. Question 4. Kidneys do not play any significant role in elimination of ammonia. Ammonia is readily soluble in water and diffuses across the body surfaces. Second, ammonia is excreted as ammonium ions through gill surface, NH2+. Question 5. The amount of filtrate formed by kidneys per minute is known as GFR. Amount of filtrate formed by kidneys per minute is known as GFR, glomerular filtration rate. In a healthy individual, GFR is approximately, this is important, 125 ml per minute. That is 180 liters per day. Look at that. What a great performance of the duty. Question 6th. Tadpole is amnotelic or aminotelic because excretion of ammonia requires a large volume of water. Since tadpole is in water, so which is the tadpole has in its surroundings. An adult frog is ureotelic because elimination of urea requires a moderate volume of water that is much less compared to ammonia. And also adult frog is amphibian. It will be there on land as well as underwater. Next, urea cycle. It is also called Krebs ornithine or ornithine cycle. So here the most important ones are the that are there in the center: arginosuccinate, arginine, then ornithine, and citrulline. These are the main things that you need to focus. So the first one you can see carbamyl phosphate, that is forming inorganic phosphate, and the ornithine becomes a citrulline, and then citrulline in the presence of ATP being broken off into ADP and inorganic phosphate, aspartate becomes arginosuccinate, and arginosuccinate becomes fumarate, and then arginine. Now here you can see substituted guanidines and agmatine. Now carbon dioxide is the byproduct that is coming out of it, and this arginine now has got to mix with water and becomes urea, and in the absence of water it becomes ornithine. Now another one is between the arginine and citrulline there is another intracycle. So arginine having this water becomes ammonia and not having the citral. Similarly, arginine having oxygen and nitric oxide will be eliminated, becomes citrulline. So you got two cycle within the cycle in this. That's why it's called Krebs ornithine cycle. And then we'll go to question number 8. Dialysis machine is also known as artificial kidney. It filters blood when the kidneys fail. In dialysis, small solute molecules diffuse through a semi-permeable membrane as a substitute for glomerulus. It has a cellophane membrane where the blood of a patient is made to flow on one side of the membrane and the surrounding fluid on the other side. The waste from the blood move into surrounding fluid through cellophane membrane. It is needed when kidney fails to work and urine is not formed. So this is a stopgap arrangement. Unless someone is ready to donate one of the kidneys to the person, of course we need to keep in mind the blood group should match. So until that gap time of a donor donating it, the person has to go for dialysis. And this is artificially created and everything is a machine one, and that helps in purification of the blood. So the urine will be eliminated through artificial dialysis. Question number 9. Urine formation involves 3 main processes. This is a long answer type question, and I told you long answer type question always you need to substantiate your answer with a diagram. That is one, fix it, label and draw. Second one is first write points. Maybe since it is 5 marks, you write 5 points, and after that each point you expand. That will suffice. So here first point, glomerular filtration. A protein-free fluid is filtered from blood of glomerular capillaries into the lumen of Bowman's capsule. The filtration occurs through 3 layers which form the filtration membrane. They are endothelium of glomerular capillaries, epithelium of Bowman's capsule, basement membrane between the 2 layers. The epithelial cells or podocytes of the Bowman's capsule are arranged in an intricate manner to leave some filtration slits. The blood is filtered so finely that the composition of the filtrate is very similar to plasma except for the plasma proteins. The glomerular filtrate rate is about 25 ml per minute. Second point, reabsorption. In fact, it should be selective reabsorption. Nearly 90% of the filtrate is reabsorbed in the renal tubule of the epithelial cells of the lining of renal tubule. Certain substances like glucose, amino acids, Na+, K+, Ca2+ are reabsorbed actively. Actively means by spending energy. Water is reabsorbed passively. No need to spend ATPs. But with the help of osmosis, water will be reabsorbed. Certain other substances like chloride ions are absorbed passively. So these are all essential for the body. That's why they need to be reabsorbed. And as I said, draw the diagram and label it. Look at that. When the arrow mark is out and the arrow mark is in, two are there. And the whole thing is taking place in the kidney. That's why cortex and medulla. So basically it's a nephron diagram. So here you go glomerulus, Bowman's capsule, proximal convoluted tubule, and distal convoluted tubule, and Henle's loop, that U-shaped structure, Henle's loop, and of course collecting that. And third step, tubular secretion. It is the process by which certain substances or ions like K+ and ammonia are directly secreted into the lumen of the nephron. The step is important in urine formation as it helps to maintain ionic balance and pH of the body fluids. So you are seeing here K+ and the ammonia are directly secreted into the lumen. So you can see here K+, it is directly secreted, and little bit of urea also you can see here in the Henle's loop, and all the important minerals, they're all here, see, they're all secreted, reabsorbed back. So overall you need to understand that the pH is to be maintained throughout, plus osmotic concentration or osmotic gradient is to be maintained. Then only urine formation takes place. So the overall thing is osmosis is very, very important in urine formation. So next session we will solve one more paper of one more chapter. Until